Good afternoon. Thank you for joining us today. Welcome to part four of our five-part webinar series. Today's topic is the drivers of change, the science of harvest. I'm Jim Morrison, the president of Hudson River Environmental Society. I'm definitely looking forward to hearing today's presentations, and we trust that you'll find the talks to be both informative and enjoyable. Today's presentations will focus on the management of our coastal and Hudson River fisheries. This symposium continues the 51-year historical mission that the founding board members of HRES had in 1970 to establish a non-advocacy organization that would provide a platform for open discussions and sharing of the environmental research data that was being actively debated during the Hudson River negotiations. HRES's current board of directors are pleased to be able to continue the original mission of HRES by providing these annual symposia to our members and friends. I want to give my personal thanks to Clay, Jim, Chuck, the Hudson River Foundation, HRES webinar teams, as well as all of our speakers who are participating in our symposium. As always, it's my pleasure now to introduce Mr. Clay Hiles, the Executive Director of the Hudson River Foundation.
Good afternoon. I add my welcome and my thanks to all of the people Jim mentioned, plus to Jim himself. The Hudson River Environmental Society has a long history of putting us all in the same room for the day once a year, and has very flexibly and creatively extended that to this 5-session symposium, which we are delighted to co-sponsor, and in which we have a special interest because we want to milk it for all it's worth in our strategic planning, our developing of research priorities for ourselves and for everybody else on the Hudson River and watershed for years and even decades to come. I hope that you will all make it to our next session. Bring your friends, because in addition to wrapping up what we've been doing and setting the stage for moving forward with what we have learned, we will have the opportunity to remember Dennis Sikowski, our science director, who stands behind so much of what we do, including the work of the symposium, and whom we miss but think of and have in our work every day. We will have that chance to pay tribute to him as part of the program next week. So welcome. Jim?
Hi, good afternoon. The seminar is being recorded, and if you miss any of the talks this afternoon, they will be available on our website by tomorrow morning, as well as all the past seminars. So we encourage you to look at that at hudsonriver.org. Our speakers are speaking 20 minutes each today, and we're going to do our best to stay on that time frame. And so we'll ask questions. We'll hold all questions until the end, approximately 3:15 this afternoon. So we ask you to ask your questions through the Q&A dialog box, and Chuck Muir and I will do our best to gather those questions and present them to the speakers at the end of the session. If you have any technical problems, please put those in the chat or the Q&A as well, and we'll try to address them. And in the unlikely event there is a major disruption, we will stop this recording and session and send out a new link shortly. We haven't had any problems, so we don't expect to today. And Chuck.
Thanks, Jim. So let me get my notes here. So good afternoon. Just a little information about the series. So the content of the 5-week series was developed by a team of scientists and resource managers, many of whom have had 2 decades or more experience studying or managing the Hudson River fish community and ecosystem. As Clay mentioned, this year we've teamed up with HRF. We have been teaming up with them a number of years to hold these, but this year is a little bit extra special. HRF has close ties with the research community and the Hudson, and they're looking to their future to develop their next strategic plan. And so by organizing this series of talks, we brought the experts together, and the Hudson River Foundation is using this group of folks now to help guide their future research on Hudson River fishes. And then, finally, this is also a dedication to Dennis Sikowski's life, who was a true friend, mentor, and colleague to many of us. So the layout has been 2-hour webinars, one per week for 5 weeks. This is the 4th one. Each session we've designed to sort of tell its own story, but certainly together you view them all, you're left with sort of a bigger understanding of the issues. And so, like Jim said, if you've missed any, you can certainly view them. They've been recorded and they're on Hudson River Foundation's site as well as the Hudson River Environmental Society's website. So thank you all for taking the time today to participate. I also want to thank the speakers for putting together today's talk. So without further delay, I'll introduce Dr. Isaac Wergen. So Ike Wergen started his education at the Hofstra University, where he got his bachelor's in political science. He moved on to receive his master's and PhD in biology from technical difficulty here. Hold on. From City College. He's currently has a professorship at the Department of Environmental Medicine at New York School of Medicine. His research interests basically 2 broad areas: population genetics of anadromous and marine fishes, and he also studies ecotoxicology of fishes with an emphasis on PCBs and dioxin effects on Hudson River fish. He's published well over 100 articles to date on those broad areas. In 2020, he received the Hudson River Environmental Society's Outstanding Environmental Research Award for the work he has done over the years. And so without further ado, Dr. Wergen, take it away.
Okay. Am I good?
Yep.
Yes.
Okay, before I start, I'd like to just acknowledge the people who did the work I'm going to be talking about. Lorraine Masita, who's been a technician in my lab for 30 plus years. Eva Hasagawa, a student who has done the striped bass work I'll show you later on. John Waldman, who actually did lab work many years ago in my lab and who's co-authored many of the papers I'll talk about, and Tim King from the USGS and Leetown Science Center, who's deceased, who is a co-author on several of the sturgeon papers I'm going to be talking about. So you've already seen this image that I projected up here of the big 3, the 3 Hudson River glamour species, Atlantic sturgeon, striped bass, and shad. And I'm going to show you some data involving stressors on these species outside of the river in coastal waters and distant estuaries. I'm going to focus on sturgeon, have a couple of slides on striped bass, and I'm going to exclude American shad, although we've done similar type studies with American shad in the past. So why do we care about these kind of things? Well, Atlantic anadromous fishes spawn in many rivers from the St. Lawrence in Canada to the St. John's in Florida. And these populations in these rivers are often reproductively isolated and genetically distinct. These populations within species could vary in their abundances and certainly in their life history characteristics, but distinct populations within each of these 6 species mix in coastal aggregations when they may be vulnerable to anthropogenic stressors. And it's important to know the vulnerability of individual populations to these stressors outside of the natal estuaries. So unlike all the talks you've heard so far, we're going to be talking about stressors today that are not within the rivers, not within the spawning rivers, but are outside of them, primarily in coastal waters. So what are some of these stressors that may impact the anadromous species we're going to be talking about? Well, obviously, they're targeted coastal fisheries, but coastal bycatch is a big topic in terms of potential stress on anadromous fish populations. In terms of the sturgeon, vessel strikes are a big deal. There may be impacts of tidal and wind power generation facilities on these anadromous species outside of their natal estuaries. And for Atlantic sturgeon, strandings actually is an issue of importance. So the way that I'm going to proceed, the way my lab works, is we look at genetic markers to identify differences between the spawning populations within a species. And then we use these markers to actually identify individual fish at sites distant from their natal estuaries, or we can actually make estimations of the contribution of the individual populations to these mixed aggregations outside of their natal estuaries. So over time, the toolbox for genetic analysis to accomplish these objectives has evolved dramatically. So back 30, 40 years ago, protein electrophoresis was the way to go, then followed by mitochondrial DNA, then microsatellites. And the talks I'm going to be, and the studies I'm going to be talking about today use microsatellites to distinguish the populations and to identify their representatives in coastal fisheries. And it's important to know that microsatellites are neutral genetic loci. They're not generally impacted by selection. And the most current technique, next generation sequencing of single nucleotide polymorphism, has even increased sensitivity over other microsatellites. And that approach utilizes neutral and selected genetic loci. So for effective management of these populations outside of their natal estuaries, it may be critical to estimate the relative percentage contribution of each reference population to the mixed assemblages. And that is called mixed stock analysis. You're coming up with a percentage of the contribution of each of the populations to the mixed assemblage, as opposed to individual-based assignment testing, in which you can actually take each fish in the mixed aggregation and identify its river of origin. So how do these approaches work? Well, the first thing you have to do is to characterize genetic variation within the reference or baseline populations that contribute to the mixed aggregation. And for that, you want to either collect very young fish, pre-migratory juvenile fish, or spawning adults on the spawning grounds at spawning time. So those are your reference individuals for the spawning populations. Then you want to characterize genetic variation in the mixed aggregations at the same genetic markers that you applied to the reference populations. And then what you want to do is you want to compare the genetic variation patterns in the mixed coastal aggregations to those of the reference baseline populations. And then what you could do is you can actually measure how accurate each of these estimates is, and it's dependent upon the degree of genetic variation among the reference populations. The greater the genetic variation that you have among your reference populations, the more accurate the mixed stock analysis and individual-based assignment testing will be. If you don't have genetic variation among your reference populations, you're not going to have very accurate estimates of the contributions to the mixed aggregations. So I'm going to start with talking about Atlantic sturgeon, because we've done a lot of work with Atlantic sturgeon in the past decade. So a little bit of relevant biology to the types of analysis I'm going to be talking about. So Atlantic sturgeon are spawned in estuaries from the St. Lawrence in Quebec to the St. Mary's River, which is on the Georgia-Florida border. And these guys are resident in their natal rivers as juveniles until they're 2 to 6 years of age, and generally below 50 centimeters total length. And then when they're subadults and adults, they become highly migratory in coastal water for extended periods of time and for great distances. And Atlantic sturgeon are listed under the Endangered Species Act as 5 distinct population segments or DPSs, of which 4 are endangered, including that in the New York Bight, and the one in the Gulf of Maine is listed as threatened. Despite the fact they are listed as distinct population segments, they're usually managed on a population-specific basis. Okay, so in terms of reference populations, we've been collecting individual juveniles or adults for many years from all of the rivers that you could see on the left-hand side, ranging from the St. Lawrence in Quebec to the Altamaha in Georgia. And you can see that we've collected many fish, looks like about 850 from the Hudson over those couple of decades. And here you can see in the third column our accuracy in assigning an individual back to the river in which it was collected in what are called leave one out tests. So you can see that we're highly successful in assigning fish back to rivers in Canada, the Hudson. When we get to rivers in the southeast, those rivers in Georgia, our accuracy in terms of assigning fish is much less than it is in the more northern rivers. So this is our baseline data. These are the baseline individuals, and over time, you can see that we collected in total and characterized about 2,500 fish. Okay, so we're going to start from north and we're going to proceed to southern populations or southern mixed aggregations. And here, let's look at some data here from the Bay of Fundy. This is the paper we published with John Waldman as I think the first author back maybe 10 years ago. So here we're looking at the origin of subadult and adult Atlantic sturgeon that were bycaught in this weir fishery you can see in the lower left-hand panel within the Minas Basin of the Bay of Fundy. And you can see that, as you expect, most of the specimens came from the St. John River in New Brunswick, with a good number from the Kennebec, but you could see that Hudson River fish migrate that far north and make a small but important contribution to bycatch as far north as the Minas Basin. And you can see a few of the fish also came from the James River in the Chesapeake Bay. Now, in a paper that we published several years ago, there were 11 Atlantic sturgeon adults that turned out to be stranded in Scotts Bay up there in the Bay of Fundy. So there was a lot of interest in determining where those fish came from, what population they originated from. And of those 11, it turns out that 4 of these adult fish assigned to the St. John River, which is the closest river to where they were stranded, but you could see that 7 of them also came from the Kennebec River within the Gulf of Maine. Now we're getting a little bit closer to home, and we're going to be talking about some specimens that were collected from Long Island Sound and the Connecticut River by Tom Savoy, who works for the state of Connecticut. And these fish were either collected from 2 sites in the eastern Long Island Sound or from the lower Connecticut River. So let's just look at the fish from the Connecticut River in the second and third column. And as you would expect, most of them, about 2/3 of them, came from the Hudson River. And if we look in the Long Island Sound, fish from those 2 sampling sites, once again, as you can expect, most of those fish came from the Hudson River also. But you could see contributions from populations as far south as the Savannah River, Georgia, and as far north as the St. John River in New Brunswick. Okay, that's as you would expect, because Hudson is very close to Long Island Sound, so you'd expect to see those specimens, the Hudson predominating the collections from those sites. But historically, there was spawning in the Hudson River, but there hasn't been any seen for the past century or so because of the construction of a number of dams on the river. So for probably close to 100 years, there was no sign of young life stages of Atlantic sturgeon in the Connecticut River until 2014. And you can see in the figure we have here, in 2014, a good number of age 1 Atlantic sturgeon were found in the Connecticut River by Tom Savoy and his co-workers. And actually one adult washed up near Lyme, Connecticut on the Hudson River. And you can see that specimen in the bottom right of the picture. So the question became, where did these young fish come from? Were they remnants of were they part of a remnant Connecticut River population? Most likely, you would think they were migrants from the Hudson, or maybe they came from someplace else. So we did mixed stock analysis on these age 1 specimens that were collected in the Connecticut in 2014. And it was shocking to see that the Hudson made no contribution to that collection of age 1 fish. And in fact, most of the age 1 fish, the greatest percentage, came from, had ancestry from the Altamaha River in Georgia, with the second biggest contributor, the James River in the Chesapeake Bay, and a small contribution from the Ogeechee River, also in Georgia. So this was a very unexpected finding, and suggested that these fish were descendants of colonists from these distant rivers. Very unexpected. Another study that we did, in this case, we looked with Dwayne Fox from Delaware State University and Kevin Walk, the commercial fisherman you can see in the picture. We analyzed large adult, these were really big fish, collected in May off the Delaware coast. And in this case, you could see that the largest number of fish came from the Hudson River, with a smaller contribution from the nearby Delaware River, with a decent contribution from the James. So this was kind of expected, because we know that the Hudson River in recent times probably supports the largest population of Atlantic sturgeon coastwide, whereas that in the nearby Delaware happens to be one of the smallest populations. So this was an expected finding. And these were primarily adult, very large adult fish, and they were all collected off the Delaware coast. So what I'm showing you here is results of bycatch these are fish that were bycaught in fisheries that extended from the Gulf of Maine, as you can see in the left-hand panel. And these fish went from the Gulf of Maine, they were collected down to Cape Hatteras, North Carolina. And these fish all came from NOAA's Observer Program, and they were collected in the mid-2000s. So this is really giving us a picture coastwide of which are the biggest contributors to the coastal fishery. And as you could see here, so this is a mixed stock analysis I'm showing the results from. So this is the percentage of fish from each of the baseline rivers that is contributing to coastal bycatch from the Gulf of Maine to Cape Hatteras. And you can see here, consistent with what people think its population size, the Hudson River was the largest contributor to this coastwide bycatch. Okay, so we could tease this data apart a little bit by using some individual-based assignment testing. So what we looked at is in terms of mortality. We know that fish that are bycaught in the sink gillnet fishery have a relatively high mortality rate of about 20%. So here we're just looking at the origin of fish that were caught in sink gillnets and exhibited mortality. And you can see that the highest mortality within the sink gillnet fishery, which was primarily for monkfish, was incurred on the Hudson River population. Okay, so this is our southernmost collection. So back, this is back 2 decades ago. These are fish that were by-caught off of the Carolina coast during the winter months. This was a trawl survey that was focused on collecting striped bass, but Atlantic sturgeon were a bycatch of this survey. So we're looking. This was this incurred during the winter months. These were fish that were provided by Wilson Laney of NOAA. And you can see here that in this case, the Hudson River contribution is not that great. About half of the bycatch of Atlantic sturgeon during the winter months off Cape Hatteras actually were fish of Chesapeake Bay origin. And the Hudson contribution is quite small. Okay, let's change gears to a different species. This is not published. Everything else I've showed you except for the winter North Carolina sturgeon was all published in journals. This is data on striped bass that were collected in 2019 from the charter boat fishery off Montauk Point, New York. We picked Montauk because it's within the migratory path of fish that are spawned in the tributaries, in the 4 major tributaries that are believed to support the coastal migratory stock of striped bass. Those being the Hudson River, Delaware River, multiple tribs of the Chesapeake and the Roanoke. And we picked Montauk because number one, it supports a large charter boat fishery, probably the largest in the state and one of the largest on the coast. And it's probably within the migratory path of all 4 of these big stocks. So historically, the study by Bergeron and Lieberman, which used 5 morphometric characters and was conducted on fish collected in the mid-1970s and is used by NOAA as their barometer of contributions of mixed stock, that study suggested that the Chesapeake contributed about 90% of fish to the mixed stock coastwide. And the Hudson contribution, it estimated, was much smaller at about 7%, with a small contribution from the Roanoke. Whereas we, using mitochondrial DNA on striped bass collected from the East Hampton Holstein fishery in, I think, 1989 or 90, we estimated the Hudson contribution as much larger at more than 70%, with a 27% Chesapeake contribution. So we used microsatellites to determine the contribution of the Hudson River, a pooled Chesapeake stock, and the Roanoke to about 410 striped bass that were collected from the charter boat fishery in 2019. And we were pretty accurate here, as you can see, in terms of our test to see how accurate we were in terms of assigning fish back to the populations from which they were collected. So you can see that for the Hudson and Chesapeake, we were over 90% accurate in terms of our assigning them back to the populations from which they were collected. We saw no differences among rivers within the Chesapeake or between the Chesapeake and Delaware. So we pooled all of our Chesapeake populations, of which there were 7, with the Delaware, and considered that as a single population. And we had the Roanoke too. And here's our estimate of contribution. So you can see that for this fishery, in 2019, it was primarily supported by specimens from the Chesapeake, with a much smaller contribution from the Hudson River and an even smaller one from the Roanoke. So in conclusion, Hudson River anadromous fishes encountered stressors, numerous stressors in coastal waters near and far from their natal river. The effects of these stressors distant from the Hudson need to be considered in management of its populations. Genetics provides a sensitive tool to quantify the numbers of individuals affected by these stressors, and contributions of individual populations to coastal aggregations and stressors varies over time and space. And that's it. Thank you.
Thank you very much, Ike. Next up, we've got Greg Kenny. So Mr. Kenny got his bachelor's degree from the University of Vermont and a master's degree from SUNY New Paltz. He's currently the Hudson River and Delaware River Fisheries Unit Leader at New York State DEC, and he is responsible for developing fisheries management plans for several recreationally and commercially important marine and estuarine fish species. So take it away, Greg.
Hi, my name is Greg Kenny. I work with the New York State Department of Environmental Conservation. I'm in the Division of Marine Resources, and I'm based in New Paltz. And my team's charged with managing the diadromous fish, the fish that spend part of their life cycle in the ocean and part of their life cycle in freshwater. We do that in both the Hudson and Delaware rivers. Quick overview of what I'm going to go through today. I'm going to give a brief summary of the past and present fisheries in the Hudson River, describe the role of the DEC and regional councils and commissions in fishery management, and then highlight some of the challenges to coastal fish management and the importance of monitoring and research. The Hudson River has a long history of commercial and recreational harvest. Humans have been using the fish of the Hudson River as long as humans have been living in the Hudson Valley. Native Americans and European settlers alike harvested the fish of the Hudson River. It wasn't until the 1800s that modern fishery management records began to be collected, and the late 1800s that rules for harvest began to be established in the Hudson River. The most popular commercial fisheries over time have been Atlantic and short-nosed sturgeon, American shad, river herring, blue crabs, striped bass, and American eel. But there was also harvest of rainbow smelt, Atlantic tomcod, white perch, and even white sucker. Unfortunately, PCB contamination and overharvest have combined to end most of these fisheries. And the only commercial fisheries that remain today in the Hudson River are for river herring, American eel, and blue crabs. But we remain hopeful that we will be able to restore many of these species to levels that can sustain harvest and are safe to eat in the future. Fishing for recreation has also been a popular pastime for over 100 years in the Hudson River. In fact, in the Angler's Guide and Tourist Gazetteer from 1885, Diamond Reef is listed as a fishing hotspot. It also indicates that you could rent a hotel for $1.50 a night and a boat for $1 a day. With those very similar prices, I don't know if that meant the boat was really nice or if the hotel was really lousy, but that was the ad. Today, according to a recent DEC angler study of the lower Hudson, the most popular recreational fishery in the Hudson River is for striped bass. People also fish for black bass, white perch, catfish, river herring, and carp. Tellingly, the third most popular target species was anything, which reveals that there's a lot more that draws people to go recreationally fishing than catching a particular fish. That same angler study also estimated that recreational fishing in the lower Hudson generated more than $48 million in economic output in 2017. So DEC role. As New York State began to manage fisheries in the late 1800s, we started to regulate harvest using tools like closing areas to harvest, implementing a lift period where nets were lifted from the water and people were not allowed to fish, and putting restrictions on the types of gear that could be legally used to harvest fish. Here's a game protector in 1920 putting out markers to designate the spawning grounds of American shad where you were not allowed to fish. And here are some game protectors in 1934 who had confiscated some illegal nets in Haverstraw Bay. Over time, it became clear that managing these diadromous fish that only spent part of their life cycle in the Hudson and much of their lives in the ocean far from New York need to be managed in a cohesive way throughout the range of habitat that they use, or the management would be ineffective. A major issue with state-by-state management was a typical tragedy of the commons. If you knew that the fish that came to your state were just going to be harvested elsewhere, if you didn't harvest them in your state, then there's no incentive for any state to reduce harvest themselves. Over time, interstate organizations were developed that seek to bring a way to get states to manage this shared resources collectively throughout the coast. The Atlantic States Marine Fisheries Commission manages Atlantic coastal fisheries that primarily take place in state waters, 0 to 3 miles from shore. The Magnuson-Stevenson Fishery Conservation and Management Act of 1976 set up a regional council system for fisheries from the EEZ out to 200 miles from the coast. So on the Atlantic coast, we have the New England, Mid-Atlantic, and South Atlantic councils. So today, DEC continues its traditional law enforcement role for enforcing seasons, gear restrictions, and closed areas. But we also have biologists who conduct surveys and analyses through the regional management bodies to provide a scientific basis for managing fish stocks. So these analyses, called stock assessments, consider things like fishing mortality, natural mortality, growth rates, the age structure, fecundity, and existing fisheries to develop models that give a picture of the condition of the fish stock in question. The information in these assessments are then used to develop or update fishery management plans for the species. And these management plans include actions that will maintain the stock within acceptable levels while allowing for harvest when that's possible, and that harvest is fairly distributed. And then implementation of those management actions is required for the coastal states where there are meaningful penalties if you don't comply. So some of the challenges with diadromous fish. The first and maybe most obvious challenge is just the sheer distance that these fish move, exposing them to potential mortality sources over thousands of square miles. For example, in 2005, we tagged 17 adult Atlantic sturgeon with satellite tags. And even in that small sample size, we found fish that moved all the way to Canada and fish that moved all the way down to Georgia. And then over that huge area, there are lots of ways for fish to die. Sticking with the sturgeon example, we know that there's directed harvest in Canada. They get caught as accidental bycatch in another number of other fisheries. They get struck and killed by boats. There's even great YouTube video of seals tossing them in the air before eating them for lunch. Considering all these possibilities, prioritizing their importance and crafting ways to manage mortality within acceptable levels becomes a very challenging task. Further, many of the species, including river herring and shad, are managed on a stock-specific basis. So, for example, when a stock assessment is done on American shad, we are looking at Hudson River specific data for all of the biological information I talked about when developing a stock assessment. So for us to be able to properly consider, for example, the impacts of whales or the midwater mackerel fishery, we need to not only know how how many total fish die from these sources, but also an estimate of how many of those fish are of the Hudson River stock origin. As I just described, for a number of fisheries, this is possible to do, but it's not easy. Finally, missing data is a challenge. Stock assessments are only as good as the data that goes into them. The DEC does conduct a number of surveys to monitor the abundance of fish in the Hudson River, which I'm going to describe shortly. But we also have some important gaps of information too, including imperfect surveys for some biological information and no regular collection of information about recreational fishing north of Haverstraw Bay. In addition to these known existing gaps, we also have new challenges. The DEC has relied on data collected by the Hudson River Biological Monitoring Program to inform stock assessments. This program was a collection of a number of surveys, including an ichthyoplankton, a trawl survey, and a beach seine survey. And unfortunately, that long-term data set funded by the utility companies on the Hudson River has recently come to an end. And if we're not able to figure out ways to continue the most important elements of these surveys, we will have poorer stock assessments in the future. So beyond the management, monitoring is very key for us to know what's going on with these species. And it's important for us to do this monitoring every year. The DEC has a variety of ongoing annual surveys to inform stock assessments and fishery management plans. For example, we have our spawning American shad and striped bass survey. That data is collected by a 500-foot haul seine. You can just make out the people pulling the net and the net itself in the water here. We have adult blueback herring and alewife survey. We catch adult spawning stock with a similar gear. We also monitor the adult alewife run in Black Creek using a fish weir and counter. We sample adult sturgeon as well as juvenile sturgeon. And we also conduct beach seine surveys, which monitor recruitment of the key ASMFC managed species, but also provide information on all of the young year fish that utilize nearshore habitats. The primary use of these data is to monitor the abundance of the stock. Are we seeing more or less adult fish, more or less young year fish? And we use that in the stock assessments. But the other data we collect can also be very valuable for management. And so I'd like to just share one example of how we used our monitoring data for management with striped bass recently. Striped bass are on the decline. Their coast-wide spawning stock biomass has been decreasing since 2004, and the Atlantic states have implemented a variety of measures to reduce mortality of the stock over the last several years. So the first step that was proposed in 2014 was to increase the minimum size limit coast-wide. We were able to use the data collected in our monitoring program to show that this may not be the best approach with spawning fish in the Hudson, given the existing regulations that were in place. So here's the size distribution data of striped bass collected in our haul seine program. Size in inches is along the x-axis. The proportion of fish in that size class is represented by the bars. Blue for males, that orange is red for females. And the yellow box outside the whole thing shows which fish could be harvested with the current regulations that were in place in 2014. So before the proposed change, you could keep any fish that was greater than 18 inches. And here is what the proposed rule at the time looked like. So same axes with the yellow box showing you what you would be able to harvest and the grayed out area showing what you couldn't harvest. So the proposed rule would only allow you to harvest fish greater than 28 inches. And as you can see from this chart, this rule would have shifted harvest in the Hudson River from both sexes to almost exclusively female fish. And since the reproductive potential of the species lies almost entirely with the female fish, we felt that this was not the best way to increase the spawning stock. So the approach that was adopted in 2015 is shown here. Instead of a minimum size limit, we adopted a slot limit where only the harvest of fish between 18 and 28 inches or greater than 40 inches was allowed. This allowed us to focus harvests on the male striped bass while maintaining the opportunity to harvest an exceptional fish. And we were able to demonstrate all of this because of the information that we collected in our regular monitoring programs. So in addition to the monitoring we do to inform our management, we also conduct research to inform that management. And here are the members of the fisheries team that conduct our research projects. Rich, Jess, Wes, and Amanda work closely with Pat Sullivan at Cornell and frequently conduct this research in collaboration with academic researchers at Cornell and other institutions. So I'd like to briefly touch on some of our ongoing research projects and then highlight an older project that has contributed to a very contemporary fish management issue. And it's another striped bass example. So for our ongoing work here, we know that shad are harvested as bycatch in a number of fisheries where the Hudson River stock is mixed in with other stocks. And it's important for management to understand just how many of those fish come from the Hudson. So we're working with the Thurkelton lab at Cornell to develop a genetic signature of American shad. We've collected samples from 14 river systems along the coast, and we'll conduct a whole genome sequencing of the samples. So this approach allows us to focus on genomic regions with large allele frequency differences when trying to differentiate stocks. Will hopefully lead to a robust stock assignment that we can then apply to shad samples that were collected in mixed stock fisheries we have questions about. We're also doing work on river herring to help us guide how we do our monitoring of river herring in smaller Hudson River tributaries. So in addition to the monitoring, we're at Black Creek. We're working with Pat and Suresh Sethi to explore other methods to monitor presence and abundance of herring in the tributary using egg mats. Another important river herring and shad question is related to the impacts of opportunities associated with existing dams and locks in the Hudson and Mohawk. We're working with Dan Stitch to do some modeling work to better understand the population level impacts of various mortality scenarios at fish passage structure in the Hudson River estuary and Hudson River and then the Mohawk. Finally, we're going to conduct a short-nosed sturgeon population estimate this year in collaboration with Drs. Kazak, Madsen, and Fox. This federally endangered Hudson River sturgeon has not had any kind of status assessment completed since the late 1990s. At the time, researchers estimated that there were about 60,000 short-nosed sturgeon in the river, and this was 400% higher than the previous estimates from the late 70s. So we are going to come up with this estimate using a new technique. This technique combines side scan imagery with acoustic tagging to estimate population size with minimal fish handling. Okay, so the final example here, I'd like to highlight some research we conducted on the Hudson River that was used to inform management decisions for striped bass. So the striped bass recreational fishery consists of a lot of fish that get caught and then released. The most recent stock assessment estimates 37.7 million fish each year, or the most recent year, got caught and released. Sometimes when you catch a fish, the hook causes wounds that become lethal to the fish. And often it's hard to know if a fish that swims off after you catch it and release it, you don't know if it's going to die from its wounds or not. So when we're talking about 37.7 million fish being released annually, it becomes really important to know how many of those released fish die and how many survived, because it may be the difference between a stock experiencing overfishing or not. So in 2001, a study took place on the Hudson River to evaluate catch and release mortality of striped bass. So 9 4,000-gallon tanks to hold fish were set up on shore. Volunteer anglers were recruited to catch fish with hook and line. And then we held them to see how many of the fish died over a period of 5 days. So some anglers used traditional J hooks and some anglers used circle hooks. Circle hooks were hypothesized to be less lethal than J hooks because they tend to hook a fish in the lip instead of being swallowed and getting hooked in the gut. And as a control group, for both of these hook types, striped bass were also captured by electrofishing and held in the same tanks. So the study found, anglers contributed 159 striped bass over 13 days in May of 2001. And the mortality estimates of fish caught by J hooks was 16%. And the mortality estimates of fish caught by circle hooks was 5.3%. So this is relevant because of, 37.7 million fish, right? This is the number that was released in the recent stock assessment. So using the estimates provided here, if they were caught with J hooks, this means that 6 million fish would have died after release. And if they were caught with the estimates here for circle hook mortality, just under 2 million fish would have died. So this study was not definitive, but it paved the way to our understanding of just how significant a problem catch and release mortality is for striped bass, and also suggested a possible solution of using circle hooks with bait. So after much additional debate, research, and continued panic from the declining striped bass numbers, the ASMFC required the use of circle hooks when fishing with bait on the entire Atlantic coast beginning this year. So only time will tell if this will be enough to stop the decline of striped bass biomass on the coast. But if the results we got in this study are any indication, hopefully this will be enough to turn the corner. So thank you. I hope that's given you both an overview of the management of fish for commercial harvest and recreation on the Hudson River, and has also provided some justification for the importance of research and monitoring for fisheries management.
Okay. Thank you, Greg. So next up, we have David Secor. Dr. Secor had his PhD at the Brooke Institute at USC in combination study there and at the Kagoshima University in Japan. He's currently a University System of Maryland Regents Professor at the University of Maryland Center for Environmental Studies. His lab's interest is how diversity in life history and migrations contribute to resilience in exploited species. Studied species include forage fishes such as menhaden and Atlantic mackerel, estuarine and coastal predators such as eels, striped bass, bluefish, black sea bass, and bluefin tuna, and endangered sturgeon. He advises the Atlantic States Marine Fisheries Commission, the Mid-Atlantic Fisheries Management Council, the Responsible Offshore Science Alliance on fisheries stock assessments and climate, as well as climate and wind energy impacts. In 2015, Dave authored the book Migration Ecology of Marine Fishes, the first on that topic since 1968. Dave's just like you all to know that Dave's favorite estuary, like many of ours, is the Hudson River, where he has spent over 30 years enjoying adventures and discoveries with colleagues and students. So with that, Dave, you can take it away.
Thank you, Chuck. I'm going to follow on some nice talks here and talk about some of the same kind of issues. Ocean bycatch, incidental catch, unintended catch. For river herrings and shad and Atlantic sturgeon, this is a result of regulations to protect those species. And for striped bass, as we'll discuss, it's because we're tending to love striped bass too much. Let's see. There we go. So start off with shads and river herrings. There are hundreds of tributaries that support river herring, blueback herring, alewives, probably fewer for American shad. Atlantic States Marine Fisheries Commission supports. Yeah, Jim, is
Dave,
there?
I'm sorry to interrupt. You're not full screen. Can you change your display setting to be full screen? Sorry about that.
Yeah. Huh.
You're on the like preview screen.
No, I'm sorry. I'm going to have to start over, I guess.
Perfect.
Okay, are you seeing the full screen or my notes?
You're good.
You're good.
Oh, okay,
Full screen.
good. All right. Thanks. So we have a lot of population structure, which has been alluded to before. And these Alosa leave all these tributaries, including the Hudson River. And there they intermingle with similar size shaped species that are prosecuted through industrial fisheries like Atlantic mackerel and Atlantic herring. And they show up in these fisheries, and historically it was a directed harvest. But in 2005, they were protected. So no more ocean directed catch of Alosa permitted, but that doesn't mean that the harvest stopped. And it's been observed, and this is all Alosa losses shown here. And you can see that they show up at a range of fleets in the coastal waters and also species, principally Atlantic mackerel and Atlantic herring. So these are observed. How much is too much? That's an ongoing question. But fortunately here, there is regulation on losses of Alosa on a coast-wide basis that the councils, as Greg just mentioned, they do curtail harvests when they become too high. This is based on historical fishing performance on directed Alosa fisheries. And it's unusual when they exceed limits, and the limits are regional, they're species and fleet specific. But in 2018, 2019, they were exceeded in a couple regions, and that forced closures of those very big, important industrial fisheries. So there is at least a tool there to curtail excessive losses due to ocean bycatch of Alosa. Atlantic sturgeon, we heard Ike's very nice presentation about mixed stock fisheries. The Hudson River Atlantic sturgeon fishery cropped up as the South Carolina one diminished, and there was a caviar meat fishery in the river, which are these open boxes here, on the order of maybe 50 to as many as several hundred adults harvested per year. But the fishery of greater concern was a monkfish fishery. It was state gill nets in New York Bight and other areas. And you see it ramps up very rapidly in the late 90s to thousands of fish. And recruitment plans were beginning to be observed in the Hudson River. Size limits were put in place to protect adult recruitment to the Hudson River, but it was probably too late. And in 1996, ASMFC posted a moratorium on all harvests in the U.S., and then this was followed by NIMS listing them and giving them protected status as endangered or threatened. I wanted to highlight a little bit that the Hudson River Foundation really mobilized science during this period, a very big effort and a big outcome in terms of conservation of Atlantic sturgeon, mobilizing scientists under Mark Bain's leadership at Cornell University. We looked at reproduction, recruitment, did pioneering work in telemetry, age growth, short-nosed sturgeon recovery. Again, some of the talk, the material that Greg covered, and looked at hatcheries rolls. And then NIMS came on with massive biotelemetry, acoustic telemetry. Thousands of Atlantic sturgeon were tagged, and they were tagged in these mixed stock areas that Ike brought up. And some of us had our own little backyard tributaries with receivers and our easy pass systems. And we started hearing sturgeon when and where we really didn't expect them. And this was a picture in 1998 where we thought sturgeon were kind of a large river spawner. So here's the Hudson and St. Lawrence. And here's where we are now. So Atlantic sturgeon is showing up in all kinds of smaller systems. And this also has allowed us to get at abundance estimates more efficiently and building to recovery plans. So a lot going on that was catalyzed initially by the Hudson River Foundation. And ASMFC has been busy with assessment work. It builds on the science. So Atlantic sturgeon are harvested in trawl fisheries, bottom trawl fisheries, but also state gillnet fisheries, as Ike indicated. And these state gillnet fisheries are important because this is where our mortality occurs. And if we start down here in this plot, you see the purple areas are where the fisheries are more intense, where they're observed by NIMS observers. And the red points are where Atlantic sturgeon are observed to be dead in the gillnet. And this tends to happen in regions that are a little bit farther offshore and where we have longer soak times, where gear is deployed for periods of greater than 24 hours. We have much higher mortality rates than when they have short soak times. That's the dynamic. Here's the New York Bight. It's a very important area for these state gillnet fisheries. We also have important areas off New England, southern New England, and then Delmarva. What is the stock mixture in these areas? I covered some of that historically or more recently. We've looked at this anew historically by looking at fin spines collected from those fisheries I showed you in the late 90s, because we didn't know that a lot of these populations even existed back then. And it turns out that even though we see the whole gamut of sturgeon populations in this mixing bowl area of the New York Bight, the Hudson River still predominates. So are too many sturgeon being taken? This was assessed in 2007 initially by ASMFC with leadership by NIMS scientist Tim Miller. And here we estimated, based on observer coverage, hundreds to over 1,000 Atlantic sturgeon were killed each year by this gillnet fishery. A more recent assessment by John Kosick and colleagues indicated it's less, and it could indicate a change in that monkfish fishery and how it's prosecuted, something maybe related to soak time. Our bycatch estimates from this assessment were about 0.4% to 9%. And this depends completely on scenarios of mixing levels, how important is the Hudson River to these coastal mixed stocks, and also the abundance level of Hudson River. How much mortality can the Hudson River population sustain itself with an additional 4% mortality? So you can see that these numbers are fairly high or even exceeding that reference point. So there is an issue here, even at the lower bycatch level in more recent times. So this demands action priority by NIMS, ASMFC. We don't have these kinds of regulations like we do for the LOSA for Atlantic sturgeon in terms of the councils or Garfield. All right, Hudson River. Well, striped bass is a little bit easier issue. We know that there are populations up and down the coast, but when we look at shell fisheries, especially in mid-Atlantic, southern New England, there's really only a few populations that count. And I covered this, but you know, the Hudson River is a very big contributor to these coastal fisheries. Various assessments historically have shown that in some years they might even be the majority contributor over the Chesapeake Bay. The other thing why striped bass is so important to Hudson is they occur in all parts of the Hudson, and there's different ways that communities interact with striped bass, whether they're interacting on small resident fish in the upper part of the Hudson River, or they're interacting with the fish that tend to use the lower estuary for most of the year, or when we interact in terms of assessment or with these large ocean-going fish that Greg Kenny just mentioned that come in and then leave just for spawning. And that's the group I want to talk about. These are, and when I want to talk about this idea of spawning runs or periods of vulnerability, we certainly know this for Pacific salmon, but not so much for striped bass. And we wanted to look at this. And so we had a very nice collaboration with New York State DEC scientists. They trained us how to use electronarcosis to implant acoustic tags in striped bass. And the Hudson River is just a marvelous system to do telemetry. It's all skinny and narrow, so we can set up lots of easy passes with telemetry receivers and get very detailed information about spawning run behaviors. This is a busy slide, but let's start over here just to demonstrate that all these circles represent detection ranges by acoustic receivers we set out, or New York State DEC has set out. And you see the whole river is wired. We have a massive observation system to look at migrations of striped bass, sturgeons, or any other kind of fish you might want to tag and put into the river. Historically, spawning surveys have focused on two parts of the Hudson River. This is where they typically found striped bass from the emergency days, Andy Canley and Kathy Atala and the work they've done. And those two areas are Athens, Catskills, Saki region up here, and then the kind of West Point, Lower Newburgh Bay region down here. Overlaying these survey areas are over 25 years of egg survey data from the utility year class reports. These are annual data fractioned across the different parts of the river. And you can see that there's two modes. There's a mode here, a little bit farther up, and then there's a mode down here in kind of Newburgh Bay area. And what we think is going on is we have spawning up here, eggs drift downstream with net current, and then down here, there's actually a retention feature near the salt front. All right, so we wanted to test whether there might be kind of fidelity to these two regions by spawners year after year. So we tagged 50 fish in 2016 in this region, together with New York State DEC, and 50 fish down in this region. I wanted to test if they came back year to year. Now, when we tag fish down here, we don't know if we're tagging them on the as they're heading up there. So what we actually did is we classified fish in 2017 and we looked for fidelity in 2018. Now, all this telemetry is giving us very fine scale migration behavior, just incredible rich data. These fish they don't directly move up and move down. They squiggle around, they stage, they do all kinds of things. And the challenge for us was to summarize this data. And my assistant looked at this and said, hey, this looks like an acoustic signal. You know, we have amplitude and then we have frequency. And so he used a speech recognition algorithm to summarize this in a cluster analysis. And when we did that, we found two dominant clusters, which is shown by this black median behavior line. So we have the first cluster, which is using the upper part of the Hudson River, and the other cluster using the lower reach of the Hudson River, and basically aligning with that traditional idea of where striped bass spawn. We classified 2017, 2018, we saw 87% fidelity by these tagged individuals back to the upper reach, and 100% of these individuals back to the lower reach. So there is fidelity year to year in terms of these spawning behaviors. This all occurs within the same spawning run, and this is just what you see with Pacific salmon. So it's a very interesting kind of behavior to observe with telemetry. The one important difference is that the upper reach striped bass arrived later and left later, and then the lower reach striped bass arrived earlier and left earlier, on the order of about 9 days. This was important because after they left and over their first calendar year of life, there was very different mortality rates. So the upper reach see very high survival through that first January, this is 2017, and the lower reach much lower survival, twofold higher mortality rates. And what we think is this is a difference in the exploitation rates that each reach suffered after it left. Now let's unpack this a little bit. How do we know these mortality rates? Well, just like in the Hudson River, where we have a lot of receivers, there are a growing number of receivers in shelf areas that allow us to look at the migrations and fates of striped bass after they leave the Hudson River. And they've been growing, and you can look from 2016 to 2017, a lot more assets. These are Bowen assets to look at wind energy impacts off Long Island and Hudson River Canyon here and Delaware, and we had one set out off Maryland. Massachusetts has been heavily engaged in telemetry for so many years. All right, so in 2016, we tagged striped bass, we classified them lower reach, upper reach. You see the lower reach leaves earlier. They migrate north all the way up to Maine, and then they come back down. Over winter off Delmarva in North Carolina, we don't have receivers there. Come back up, go back down. Striped bass are birds. Summering area, wintering area, breeding area. Just remarkable to see this kind of faithful migration behavior year after year. All right. So this is the kind of migration we're seeing. Two different groups spawning in the Hudson River, leaving, going up to Massachusetts, some going up to Maine, coming back to overwinter. We can set up a listening station or a detection station here, and you can get this from a Olarian perspective. Here they are coming up in spring, so the spring spawning run begins all the way down in Delmarva for a Hudson River fish, and then here they're coming back for their southern migration. Now this gets to Greg Kenny's discussion. Here's our size limit in the Hudson River designed to protect those spawners. Once they leave the Hudson River, all bets are off. Now the limit's greater than 28 inches. So what we're suggesting is a huge latent effort demand. And if you leave early, you get whammed harder in terms of exploitation. We think this is the reason for this difference in mortality rates of the early versus the late spawn run. But there's a bigger issue here as well. As Greg Kenny mentioned, this is landing's history, and we're now in this phase where the biggest source of mortality is recreational release mortality. 2019, 32 million brought to the boat. For every single fish retained, 13 were released. That's a huge increase from 2010, where for every retained, there was 5 released. Why are so many being released? Size limits are a big part of it, how these fish is regulated, desire to catch the biggest fish, desire to catch fish as much. So this is a conversation that we need to have with our recreational charter stakeholders. Summarizing, American shad river herring is a wicked problem of industrial fishery interactions with complex stock structure, even low levels. Ocean bycatch could curtail recovery because these are industrial volume fisheries. Still, there are efforts. There is action that can be tinkered with to improve status and to hopefully down the road link Hudson River populations of shad and herring to ocean bycatch. Atlantic sturgeon, we actually have the tools to do all this, but we don't really have a regulatory mechanism to enforce any limits to those fisheries that take Atlantic sturgeon if they are taking too many of them. And for striped bass, we're kind of loving striped bass too much these days in terms of the level of effort and catch in our recreational fisheries. And this could have an impact on Hudson River in terms of those two contingents, the upper and lower reach. Why would it matter that the lower reach contingent is seeing higher exploitation? Well, it has to do with their spawning. And if they're spawning earlier, and earlier spawned eggs do better than later spawned eggs over a set of environments, that could have an effect on their resilience and stability. This is now our biggest issue for striped bass, and the work, like we saw Greg Kenny present on hook and release mortality, needs to be done in many other systems. I always want to thank my assistant, Michael Bryan, New York State DEC collaborators, Jess Best and Amanda Higgs, my longtime friend, collaborator, colleague, mentor, Steve Knack, who has endured patiently all our misadventures and discoveries on the Hudson River. Thank you.
Okay, thank you, Dave. And I'm going to move on to Olaf Jensen. Dr. Jensen received his bachelor's in biology and sociology from Cornell, his master's in science and marine in marine science from University of Maryland and his PhD in limnology from University of Wisconsin. Dr. Jensen recently moved from the Rutgers Institute of Marine and Coastal Sciences to the University of Wisconsin, where he is currently an associate professor at the Center for Limnology. His research focus is on fisheries management and population dynamics, and he takes basically a social ecological system perspective with his research focusing on fisheries biology, as well as those who manage and harvest the resource. And he is joining us from the still frozen Midwest, where he's expecting about 3 inches of snow today, even though it's actually quite cold here in New York today. It's well below zero here, too. So anyway, Olaf, take it away.
Thank you, Chuck. I'll start out here by acknowledging that my goal here today is not to inform, but to provoke. We've had some very, very informative presentations by a group of panelists who know the Hudson River and its fisheries far better than I do. And I suspect that many of the folks in the audience also do. So instead, what I would like to do is to take an iconic species from the Hudson, the striped bass, and try to put it in a broader context, and to suggest that, like generals, fisheries managers and scientists may be fighting the last war when it comes to fishery management, and that there are some new challenges and trade-offs on the horizon. So it wasn't supposed to be this way. We were supposed to be successful with striped bass, and in fact we were. If we look back at 20 years ago, striped bass were the poster child of successful fishery management. We were collectively patting ourselves on the back about how well we did in recovering striped bass. And in fact, it was true at the time. If we look at a graph here of the CPUE averaged along the Atlantic coast for striped bass, the really intensive fishery management restrictions started coming in the mid-'80s, including complete moratoria in many states, driven by the Striped Bass Conservation Act. And it looked good, right? We were doing well. By 1995, the last day of data here, you could look at this and call it a success. It was almost like a movie. And if we think of fishery management as the fishery managers being the captain of their ship, they've got a hand on the tiller, which is the fishing mortality rates. And they can pull that tiller back and forth and adjust fishing mortality rates, and the whole ship of the fisheries social ecological system responds and goes where we want it to, until the tiller stops being responsive. That is, until the fishery stops being responsive to changes in fishing mortality. And in fact, 1995 might be remembered for striped bass as the last year we were happy. Since then, the recovery has stagnated. At this point, we seem to think it's stagnated below the target for recovery. And recruitment, the yellow line here, has been on a decades-long decline, with spawning stock biomass also declining over the past decade or so. No longer quite the movie we thought it was. This is not a pattern that's unique to striped bass. If we zoom out and look a little more broadly at fisheries in the mid-Atlantic and Northeast US, the same set of graphs could very well fit just a few years later for summer flounder. So same pattern, different fish. With summer flounder, we hit the bottom similarly in the early 1990s. Instituted a set of strong restrictions on fishing mortality and saw the population recover. Similarly to striped bass, we then saw a slow decline in spawning stock biomass, coupled with a long-term decline in recruitment. And if we think even more broadly beyond those two examples to the entire US East Coast, what we've seen in recent decades since the early 1990s is a pattern of declining fishing pressure. So on the left-hand side here, we have the fishing mortality rate. That's the relative harvest relative to the target, that UMSY. That's been declining since the 1990s, the early 1990s, and catch has gone along with it here. But biomass recovery has stalled. So since the early 2000s, we've seen no increases, no continued increases on average when we aggregate across all managed fisheries on the East Coast. No recovery in biomass here since the early 2000s. At the same time, many of the benefits that we hope to get from fisheries have been in long-term decline, if not free fall. Commercial landings for the Mid-Atlantic and the Northeast as a whole continue to decline. And this is, of course, an issue because our seafood consumption has not declined over this period, except potentially for the last year during COVID, which is strange and still somewhat unknown. And so what this means is increased reliance on harvest from foreign fisheries, foreign aquaculture, which is almost certainly a bigger environmental impact than well-managed sustainable seafood from the US. The picture on the recreational side is similar, if not a bit worse, in terms of the landed component of the recreational seafood. We think of recreational fisheries, as the name suggests, as recreation, but it is also a source of food, and a decreasingly important source of food here in the Northeast US. I want to point out the citation there to the State of the Ecosystem report for the Mid-Atlantic. This is a fascinating resource, and I definitely encourage you all to check this out, to get a 30,000-foot view of the status of the Northeast US shelf ecosystem. Really great work by Sarah Geiches and folks at the Northeast Fisheries Science Center. And there's cultural loss that has gone along with this as well, right? If you wanted to see the Long Island Hall seine fishery for striped bass, I would recommend a book by Peter Matheson, which documents that fishery much in the same way that paleontologists document dinosaurs. These are things long gone. And of course, they didn't go quietly. There was protest and civil disobedience associated with the end of this culture, but this unique culture is gone. And the recreational sector as well, again, is a source of discontent. I'll draw an example here from New Jersey, in part because that's where I've worked for the last 10 years. But also because if you want to know about fishermen's opinions, New Jersey is a great place to study them, because there's very little hesitation in sharing opinions among New Jersey fishermen. You never wonder if something's bothering them. And so this is a quote from one of our focus groups. What I've observed here is just absolute total frustration bordering on anger. This is in the context of fishery regulations. And this is not surprising if we look at the pattern of seasonal openings, just as one example of regulation for 4 popular bottom fish species in New Jersey. What we see is an increasing fragmentation of the seasons. We've got shorter seasons, non-overlapping seasons, just an overall decline in recreational fishing opportunity. And that's something to pay attention to, because we live in a democracy, and you cannot have a situation that goes on forever where fishermen are unhappy about the fishery management system without changes coming about that might actually end up undermining the successful aspects of that fishery management system. And one really good example of that is New Jersey's 2017 end run around the ASMFC when it comes to summer flounder management. So the ASMFC allows states to propose alternative fishing regulations that achieve the same target catch. This is conservation equivalency. It's a very successful approach in general to allow states flexibility to achieve the harvest targets in as economically and socially benign a form as possible. New Jersey submitted a proposal for conservation equivalency for summer flounder, and that proposal was rejected by the technical committee. So far, this is fairly normal. The process was proceeding as it's supposed to. In this case, it was rejected by the technical committee because the proposed regulations, there was limited evidence that they would achieve the goals, and they were relying on an assumption that circle hooks in this case would be sufficient to achieve reduced bycatch mortality. It got weirder then when New Jersey decided to go ahead with the proposed regulations anyway. So despite being told by the ASMFC that this was not approved, they decided that, well, thank you very much, we're going to do it anyway. Eventually the Secretary of Commerce, a political appointee, rejected the ASMFC's finding that New Jersey was out of compliance. This might seem like a provincial squabble in fishery management, but I think this is a sign of the potential unraveling of the social compact around fishery management, which is that all states are bound together in this agreement, and that they will solve their problems together, because we don't have New Jersey striped bass or New York striped bass. We have a migratory population that is mixing in the ocean and mixing in fisheries. I will say that the same kinds of pressures for summer flounder are also playing out to some extent with striped bass. Dave Secor's presentation mentioned that for every every striped bass retained in the recreational fishery, 13 are released. And that's led to a situation where the striped bass fishery is essentially mostly a catch and release fishery at this point. Now, that might be somewhat okay for striped bass in a way that's not true for summer flounder, because there's a strong catch-release ethic in striped bass recreational fishing, not shared by all anglers, but shared by many, whereas summer flounder are a meat fishery. People go fishing for summer flounder because they want to bring them home and eat them. So some differences here as well. And in terms of the recreational sector in general for the Mid-Atlantic and the trends, what we've seen is a recent reversal starting about 2010 in the decades-long increase in recreational effort. Still a little bit early to say whether that's a distinct new trend or not, but certainly the recreational effort is no longer rising. Even more worrisome, the recreational fleet diversity has been on a long-term decline. And what this means is that we have been losing the charter and party boat sector of the recreational fishery. The folks who make their livings catering to recreational fishermen, taking them out to fish, they are going under at a fairly rapid pace. This past year certainly didn't help with that for the charter and party boat sector. Now, it may definitely turn out that overall effort was still quite high this past year, but social distancing on a six-pack charter is often quite tricky. So what's happening? What is the explanation for this? These apparent declines that seem unrelated to fishing mortality? Well, anytime you've got a broad pattern that's shared among stocks, climate change is an obvious candidate, and certainly could be an important factor here. It's too soon to say for sure what the exact effects of climate change are. We know climate change is shifting fish spatial distributions. And an interesting paper here by Kristen Kleisner and colleagues looked at projected changes in thermal habitat with climate change. And most of the mid-Atlantic species are projected to have an increase in thermal habitat area, including striped bass here. So not obvious that climate change will, on average, on net, be a bad thing for population dynamics for many of our species. It's certainly changing things, though. If you want to go to the hot spot for recreational striped bass fishing, it's not the bridge pilings on the Chesapeake Bay Bridge anymore. It's not somewhere on the Hudson. It is the Miramichi River in New Brunswick. And so, some evidence that these populations more toward the northern end of the range are doing well, although that evidence is mixed. The St. Lawrence River populations seemingly not benefiting from the same environmental phenomenon. At the southern edge here, one of our best long-term data sets, the Maryland Juvenile SANE survey, shows the same kind of pattern in the overall population data, a decline since the mid-1990s. And in terms of climate change effects on overall productivity, I would point to a meta-analysis here that Chris Free in my lab did, where one of the only species in the mid-Atlantic that we were able to look at that had a clear signal of climate change on productivity was the black sea bass, and that signal was positive, where in warmer years, these redder circles here, the productivity at a given biomass was higher than in colder years. Maybe it's food. So of course, there's lots of discussion about whether menhaden fisheries, for example, are contributing to declining productivity of striped bass. The trends certainly don't line up here. Menhaden populations have been relatively stable since the early 1990s. If we look at other species, certainly shad and river herring have declined, but most of those declines happened long before we saw these apparent declines in productivity of striped bass. I'll just point out a paper that's coming out soon from my lab looking at marine predator populations. We found very little evidence of clear benefits in productivity for predator populations as their prey rise, assuming that their prey are kept at least at maximum sustainable yield. Okay, so to wrap up here, I think the primary fishery management challenge of the 80s and 90s was almost certainly to end overfishing and to recover our many depleted stocks. That is no longer the primary fishery management challenge. It's still important, of course. But today, I would argue that maintaining our stock recoveries and maintaining the legitimacy of the fishery management process are primary challenges. And I think the summer flounder recovery shows that well. If you were just to look at the left-hand graph here, you might say that was a success. Very few people involved in the fishery would call summer flounder a success story. And that's because the promise was always that if you take, and this was sometimes explicit but often implied, was that if you take cuts today, if fishermen take restrictions today, they will benefit in the future with relaxed regulations and the ability to harvest more and more abundant fish. Well, the fish are more abundant, but the regulations have not relaxed, and the ability to access those fish has not come through. I'll stop there, and I hope we'll have some discussion. Thank you.
Okay, thank you very much, Olaf. So next up, we have Yong Chen. Dr. Chen received his PhD in zoology and fisheries population dynamics and statistics from the University of Toronto in Canada. He's currently a professor of fisheries science in the School of Marine Sciences at the University of Maine. His research focuses primarily on the development and application of novel approaches for fisheries population and ecosystem monitoring, assessment, and management. He's received the University of Maine Presidential Research and Creative Achievement Award and the University of Maine Alumni Association Faculty Excellence Award. He received the University of Maine Faculty Mentoring Award also in a couple different years, 2 different years. He's served on the New England Fisheries Management Council Scientific and Statistical Committee since 2011. He has been the editor-in-chief of the Canadian Journal of Fisheries and Aquatic Sciences since 2013. And Dr. Chen will be coming to town. So though Dr. Jensen has migrated to the Midwest, Dr. Chen will be migrating to Stony Brook University this coming summer to work as a professor of marine science in the School of Marine and Atmospheric Sciences. One of his key responsibilities at Stony Brook will be to curate the historical Hudson River Biological Monitoring Program data and samples that have recently been donated to Stony Brook. And with that, Dr. Chen, you can take it away. And don't forget to unmute your mic, Dr. Chen.
Can you see me? Can you see the slides?
Yes.
Yeah.
Yes.
Well, thank you very much, Chuck, for the really nice introduction. And I'll get on the topic right away. So the topic I want to cover today is how we can incorporate climate-induced change into fisheries monitoring, assessment, and management. So I'm going to cover 3 topics here, and the first one would be a challenge of changing an ecosystem for fisheries monitoring, assessment, and management. And then I'm going to talk a little bit about Hudson River Biological Monitoring Program. I think Kenny talked some of that, so that saved some of my time. And finally, I want to talk about challenge and opportunities in monitoring and assessing a changing Hudson River ecosystem. So I will start with a quick kind of introduction of a decision-making process in fisheries management. So National Standard 2 actually requires us use the best available science for fisheries management. And this requires we collect high quality of data from various monitoring programs targeting recreational commercial fisheries and fish population, and also their abiotic environment. And we then use that data for conducting stock assessment and to estimate key fisheries parameters. And then stock assessment are then peer reviewed for the quality before they are used in developing management regulations. So clearly you see that it is really important to collect the high quality of data. And well, the traditional fisheries management actually focus on the regulation of a fish stock biomass and the fish mortality rate only, and with very limited and almost no consideration of a directional change in the ecosystem. And when I say directional change, I mean the change is not random, and it has a trend over time. When that happened, and like climate change impact, for example, and this slide show you the potential impact for climate change on physical, chemical environment, and the biological resources, and the socioeconomic system. And to mitigate it, you see a lot of different indicators here, and those are be affected by climate change. And to mitigate a possible negative impact, we needed to develop an adaptive approach, which usually included improve research and forecast capacity, and increasing resilience and flexibility in the management, and identify new opportunities. Although we have a very good understanding in terms of climate change impact, we actually rarely incorporate that changes into stock assessment. This is a study done in 2016. And it reviewed over 1,200 marine fish stock assessment, and they only find 24 of them actually incorporated ecological drivers. So you see that there's a big gap there. And I will use a couple of examples to illustrate how we can incorporate climate change impact in the fisheries into assessment. And this slide shows you a larger change in the habitat quality of American lobster in a coastal Maine. And this moving piece is for Penobscot Bay area, which is one of the most productive area for American lobster. The red color here, of course, suggests a high quality habitat, and the blue means a low habitat quality. You can see that habitat quality changed over time and over space. And this low panel here show you that the temporal change in habitat quality, and the red means an increase over time. And you can see that habitat along almost all coast of Maine improved over time. This slide shows you the spatial temporal dynamics of lobster distribution along the coast of Maine, and a larger change that occurred over space and over time. And the population center actually move towards the northeast over time. And this is a kind of a busy slides here, and to show you the projected change for lobster and the scallop habitat quality in the next 80 years, and with different type of climate change scenarios. And we using what we call an ensemble modeling approach for 4 areas, and see how lobster and scallop habitat quality change. And those areas include Gulf of Maine, Georges Bank, and Gulf of Maine near shore, and southern New England, and near shore. And you see that the general pattern is that habitat quality is stable or improved in Gulf of Maine, Georges Bank, but tends to become worse in southern New England for both species. And based on those kind of analysis, and we will be able to quantify the temporal change in habitat quality. And you can see that the red means high proportion of a good quality of a habitat. And you see that over time that quality change. And by assuming that better quality habitat quality will likely lead to improved recruitment, and we can link a temporal change in habitat quality to recruitment dynamics. And if we compare stock assessment with and without incorporating habitat quality change, and you're going to see the differences between these two. The top one, assessment without considering habitat quality change, and the bottom one with the incorporation of a change in habitat. And you see that the recruitment estimated in the bottom part actually is much more optimistic. So that means that we incorporated improved habitat over time. And this diagram actually shows that change in the thermal habitat actually also changes the performance of monitoring program. And if you look at this abundance index, how that changed over time, and this is actually reflect the both population change and improved catchability in the survey. So that means when they're getting warmer, and the survey can more easily pick up the lobster. So if you want to use this abundance index in assessment, you potentially are going to overestimate the population size because the thermal habitat change improve the catchability in the survey program. We also help Atlantic State Environmental Commission to develop a new assessment model that allowed us to incorporate a change in the temperature in assessment of Gulf of Maine and northern shrimp. So that's another model, but I don't have time to provide details here. This slide shows you actually our analysis of effectiveness for a monitoring program in monitoring lobster shell disease in the Long Island Sound. And what we find here is that the shell disease gravity center actually shifted over time, and you see that start 205, 201, 205 is here, but 211, 213 is here. So if you look at if you look at this diagram here, this most likely this is where the high incidence of a shell disease, but the current monitoring program only cover this areas. So that we're going to miss a big area if we don't modify this monitoring program. Now, I'd like to talk a little bit about a Hudson River biological monitoring program. And although I'm relatively new, and I just started to work on this a few months ago, but I guess I need to cover this because this is a Hudson River seminar series. And this program, consists of quite a few sampling programs, with most of them actually starting in 1974. And they divided into a different type, 3 or 4 type of different habitats, and also divided into 13 regions. So it's a stratified random survey. And the data collected in the program actually provide us with great opportunity to examine and how Hudson River system changes over time. For example, we can use the data to evaluate the change in the habitat. This is diagram show you that the river-wide water temperature actually increased by 0.3 degrees Celsius per decade. And you're going to see that it changed, and also that change is across the whole areas. And also river-wide mean dissolved oxygen decreased over time, and you can see that is also across all regions. And this one show you that there's no change in salinity over time. And climate change also resulting in a more frequent storm, abrupt change in temperature and water flow. And the figure here on the right side show estimated Atlantic tomcod abundance in different survey weeks and areas. So if you look at the top one in 1999, in July and August, and the temperature in many areas are higher than 27 degrees Celsius. So this is a red area here, right? Red area here, and you don't see any tomcod exist in those areas, and that's very different if you look at what we have in year 2000, when you see tomcod were widely distributed over all the areas, and most areas that there's no temperature higher than 27 degrees. And we have heard a lot in the seminar series, talk about it, climate change. So I'm not going to repeat it, and just want to mention that one study that I think it's really interesting, that it try to project that in the future American shed and striped bass, and how their spawning season may change it. So this study concluded that spawning season of both species were initially early in the year, and the spawning duration will be reduced over time. And this is, of course, will have a potential impact on the monitoring program in the future. Now, I really appreciate that Stony Brook actually allowed me to start to, and my student to have access to the Hudson River Biological Monitoring Program database before I even started my position. And we had some kind of preliminary evaluation of the data. And this slide show you this some of the work done so far. This is to show you for American eel and how a young of year on the top and a year and older on the bottom, and how that distribution changed and over from 70s, 80, 90, and 2000, 2010, how that changed. And you're going to see that there's a change over time and over space. And this is for American shed. And also you can see the spatial temporal changes. And this is for striped bass. And you see that it also changed. And Atlantic sturgeon, and also change it. Atlantic tomcod, and you change it also pretty obvious. So with those changes, I think, given all those changes, I think it's really important for us to think about it, and how can we address those challenges? And it is really important, I think, to continue monitoring the Hudson River ecosystem dynamics and to understand what has changed. Right, so we need to continue making observations, and we also needed to study a system to understand why those observation changes occurred. So that's a requirement for research, and then we do need to develop modeling capacity to project future change. And so the observation here, observation, research, and projection are critical for us to develop management strategy to mitigate negative impact of climate changes. So this is really can help us make a decision, a right decision to reduce negative impact of climate change. So this is a kind of a, given all those changes, I think it's really important for us to look back and look at the design that we have been using for so many years, in monitoring Hudson River ecosystem to see whether those designs still can capture what we want. And so this is a kind of a diagram show how a computer simulation study can be done to evaluate the effectiveness of monitoring program. And also this kind of study will allow us to put together a kind of integrated, develop integrated monitoring program so that we can have a more cost-effective way to monitor how the population change and ecosystem changes over time. And I think it's also really important for us to, in the Hudson River ecosystem, for single species, for example, I think it would be great if we can develop, management strategy, evaluation strategy to look at the framework, to look at the how, answer what if questions to understand, possible consequences given climate change and possible change in management strategy. And this is for kind of for population management. But if we want to look at the ecosystem level, and we may want to develop end-to-end ecosystem model that incorporate a physical biological environment and also the fisheries management in those models. And those type of models really, would be a really nice tool for us to use to address the questions that can be asked by stakeholders. And we can use those kind of tools to engage stakeholders in decision-making process, and also the results derived from this kind of study can be used to communicate the information to public to raise their awareness. So I think this is the type of tools would be extremely important in the future for us to look at it Finally, I think I want to thank SOMAS of Stony Brook, and thank my graduate student and Hudson River Foundation in New York DEC. And after all, the energy, the company that started this, gave this Hudson River Biological Monitoring Program database and the samples to Stony Brook. That gave me an opportunity to actually move to Stony Brook to work on this very valuable data and samples. And that we have a lot of challenges ahead of us, but at the same time, I think we also have a lot of opportunities. So I think I'll stop here. Thank you.
Okay, thank you very much, Dr. Chen. So Jim and I are going to just go through some of the questions, and I will start off. So Ike, first couple questions are for you. First is a question on the method when you're sampling Atlantic sturgeon to do the genetic work. Are they sacrificed, or are the fish caught, sampled, and released?
No, they're not sacrificed. We take fin clips, very small fin clips, and put them in alcohol. And that's a great source of DNA.
And then the second question, do you have any data on the Atlantic sturgeon in the Merrimack River in Massachusetts?
No, I think I don't have any data. I may have gotten some samples a long time ago, but I don't remember what we did with them. I don't think there's any spawning there, so we certainly would not have used them as a reference population.
I'll go to Greg Kenny. Question from Marsha Johnson. She's wondering, in terms of managing shad, sturgeon, and striped bass, what effects do you think the recovery of the menhaden might have?
Yeah, it's an interesting question, and I'm immediately thinking of Olaf's graph there that has kind of shown that the abundances have been higher for quite some time now already. And thinking about Yang's work projecting into the future. But I guess briefly, I think it would be most likely that they would change striped bass, being a prey item. As far as the other elocids, I'm not immediately thinking that there'd be much relationship between that. So those are my initial thoughts.
Maybe one more. This actually comes from Chuck. Do you anticipate climate change will make it more challenging to manage Hudson River fisheries? And if so, how do you incorporate the effects and management recommendations? Softball
Oh
question.
yeah, climate change, that should be easy, right? Yeah, and I think we're hopefully, it's great we have Yang now in New York, who does these kind of projections, for fisheries management. And, we could hopefully use a lot of the work that he does to help inform those stock assessments moving forward, just like he did with lobster.
Okay. Next, have a question for Dave Secor. This is from Jeff Leventon. Is there a Hudson-specific management strategy that might encourage shifts in spawning sites to maximize spawning success and minimize mortality?
Yeah, well, I don't think there's a Hudson River specific strategy because the mortality is happening outside of the Hudson River currently. And as Greg indicated, it used to be, the reason we did that study initially was there was very high mortality that was indicated in the Hudson. When they did the slot limit, we didn't see it. So in order to protect both those spawning contingents, you would have to use some kind of area season regulation or change your size limits so that they aligned with what was going on in the Hudson River.
All right, and then one I had a little curious about, for PCBs and striped bass, we're finding striped bass that are caught upriver have much higher concentration of PCBs than downriver. And just seeing the difference in mortality rates there, you have a higher mortality rate on striped bass on the lower river versus the upper. Could that somehow factor into the reason why we're finding these higher PCBs in these in the fish that are caught upriver?
Yeah, I don't think so, Chuck. A generation ago when I started research on the Hudson River Foundation, PCBs were a big part of their research program. And we discovered that there are resident freshwater fish that you might encounter in the fall that were just loaded with PCBs. And we use congeners, in fact, to also, and otolith chemistry to show that resident behavior was linked to PCB contamination. The upper reach striped bass, they're the same migratory ocean contingent that moves in and out seasonally as the lower reach group. And I wouldn't expect to see differences in PCB congeners or levels.
So I'll go to Olaf. Jim Turk asks, do you know whether or with certainty that the recreational effort is truly declining as opposed to more recreational fishing pressure associated with private boats and less so from charter and head boats?
I don't think we know for certainty anything with respect to the recreational fishery, just because it's not nearly as well monitored. But the shift from party charter to private anglers should be reflected in that data. The data that I showed comes from the State of the Ecosystem report, and the original source is the MRIP program for estimating recreational effort and landings. So it should have accounted for that. Whether it did with accuracy, I think is hard to say. There have been recent recalibrations of MRIP that I think have left many people skeptical of it. Even before that, when it was MRFs, obviously a lot of skepticism about the ability to survey a vast and diffuse angler population already.
Great. Thanks. Just one more. As climate change shifts northward, do you think you'll see a shift from the local anglers to target different species? Sorry about that. And if so, what can be done? What can local government people do to try to encourage that shift? that make sense? Sorry.
Yeah, I think, is that for me, Jim?
Yeah.
Yeah, I think there's already plenty of that going on, and black sea bass and summer flounder are prime examples where they've expanded their range and are now being caught in places that either didn't have them historically or didn't have important recreational fisheries for them historically. Black sea bass now off of Rhode Island, for example, they used to be occasionally found there, but now they're a big fishery. And important issues now with respect to the state-by-state allocations to the quota. As the species have shifted, the quota has not shifted, keeping pace. And so there are big debates right now among the states in how to reallocate that quota or whether to do it.
Great, thanks.
Next question is for Yang. It's from Karin Lindberg. What factors drove the change in lobster catchability that you presented, and was there habitat compression, and if so, what was the cause of it?
So lobster population kind of exploded over the last 20-something years, and so that lobster that appeared in a habitat that's traditionally not their favorable habitat. And so they are kind of widely distributed, and usually they stay in a very complex habitat that has very limited access to trawl survey. And so with kind of a widespread distributions and take on different type of habitat, not even non-traditional habitat for them, and you have to actually, the lobster trawl survey actually catch more, not lobster trawl survey, it's the bottom trawl survey actually catch more than they should be in the past, compared with the past. So that you see the catchability increase over time. So it's more like a kind of a change in kind of distribution patterns because of a change in the thermal habitat and increase in the population size. So both independent and density dependent and density independent factor actually regulate this kind of changes in survey catchability.
So, and the last question, I'll start with you, Yang, and then anybody else has some thoughts on this. Is sort of a thinking, a more philosophical. It's from Vernon Benjamin. And is there a basic assumption that improving fisheries management will have benefits for controlling climate change?
This is for me, or?
It's for anybody who would want to take it. If you have a thought on basically fisheries management related to controlling climate change, and is there
I'll jump in, Chuck.
in doing so? Yeah.
There was a nice paper a couple of years ago by Steve Gaines and company out at UC Santa Barbara on this at the global level, showing that a lot of at least the projected short-term impacts of climate change could be offset by better fishery management. If we simply globally stop overfishing for many stocks, that would offset on net the effects of climate change. Of course, at any local level, it's much more complex than that.
And I think it's also important to increase the flexibility in management regulations. And I think a lot of time, we take a long time to do the stock assessment and then make a decisions. And once we made a decision, it's hard to make a change. I think if we can increase the flexibility in that decision-making process and have some rooms there for a potential change that we're not seeing during the assessment, that will probably accommodate some of the changes that are needed.
Great. Well, thank you. Jim Lodge, do you have any last comments?
No, I think everyone did a great job today. I just want to thank all our speakers and for all of the participants, and welcome you back again next week. Hopefully, next Monday for our final series. But thanks again for staying with us.
Okay. Thank you.
Thanks, guys.